Hello, everyone. Hello, everyone. Welcome to session 8 of BGA24. Today, we're joined with the GRIT team, who are going to be taking you through an introduction to manual curation. Please take it away, Dom.
Oh, thanks, Damon. I take it everyone can see the slides okay. Cool. Thanks, Damon. Cool, yeah, so welcome to our workshop on manual curation, predominantly using HiC. We ran this last year, so if you're here for a refresher, welcome back. And if you're new, just welcome. The session's being run by, as you can see, the Genome Reference Informatics Team here at the Sanger Institute. We delightfully call ourselves GRIT for short. So my name's Dom. I'm one of the curators here. So I'm going to run through an introduction to curation, what curation is, and why we do it. And then we're going to have a bit of a practical session where two of the other curators are going to run you through the actual process of performing a manual curation. So Joe number one, Joe Collins, is going to do a walkthrough of a map rearrangement. And then Joe number two is going to do a run through of the scripts that we've run to then process that manual curation and output our nice, neat, curated fasta. I've been asked to ask you if you could start launching Gitpod now, because sometimes for some people it can take a little while to get going. So whilst I'm giving this presentation, just try getting that going in the background, and hopefully there are no issues. We have had a couple of issues, so if you do have any issues, just let us know. Whilst we're on that, it's most helpful for us if you have questions to put that in the Discord. There are a number of the curation team here present today who are keeping an eye on the Discord channel to answer any questions. So feel free to just fire away questions to them whilst any one of us are presenting today, and one of the other team members can pick up your questions and answer them for you. So what is genocuration? We're basically the process of taking raw assemblies, genome assemblies, and taking all available data and elevating that assembly to essentially chromosome level resolved assemblies. And we use data types predominantly HiC, but we also use karyotyping information. And we're also trying to remove any traces of contamination that may not have been picked up in the decontamination process. And we're also removing haplotypic duplication, because generally speaking, when we're producing an assembly, we're producing just a single haplotype. And sometimes we can retain two copies of stuff in that single haplotype assembly, which is less than desirable, so we try and remove that. And the last little phrase at the end there is in a reasonable time frame. Now, if any of you have ever seen a HiC map or worked in Pretext, let me tell you, you can spend hours and hours, if not days, weeks, months, poring over these maps and making tiny little adjustments, which is fine if you're in your research group and this is your baby genome. and you're really precious about it, spend as long as you like. For us here at Sanger in the Tree of Life project, we're trying to get through as many of these curations as quickly as possible, because we're trying to hit high numbers of outputted genomes. So this reasonable time frame phrase is really important to us. So we have worked on a number of different projects over the last few years. Our main project being the Darwin Tree of Life project, for which we're closing in on around 2,000 outputted genomes now. We've also worked on the Vertebrate Genome Project, aquatic symbioses, the European Reference Genome Atlas, and various other projects. So we have quite a good bank of knowledge on doing manual curations now. This is the general workflow that we use here at Sanger. And so you can see, you may be familiar with this process. You know, the samples come in, they're barcoded to validate the species identification. Then we do the DNA extraction, DNA extraction and sequencing. We're using long read sequencing, HiFi, PacBio. We're also doing HiC sequencing, which is the crucial step for the manual curation. That data then goes through our assembly team. It all gets assembled and comes to the curation team, where we work our magic, do a manual curation, and hopefully output chromosome level assembly to the public, and ultimately a publication of the genome note. So what we're talking about today is this step of the process, the curation process. And as I was saying, we're the last step before this data becomes public. So we're really the gatekeepers for quality of the assemblies that become available to the public, and we want them to be really as high quality as possible. So we're really doing everything we can during manual curation to ensure that they're as correct as they can be given the data that we have available to us. And this is necessary because although the quality of the algorithms and the assembly process and the data itself is becoming better and better every year, we're still seeing issues every day. We see problems in order and orientation, scaffolding issues. We have misjoined assemblies, false duplications or technical duplications where the assemblers literally copy and pasted a sequence. And then we also have just quirks of biology. So, for example, birds have microchromosomes. They're very hard to assemble. They're very small, highly repetitive, and it takes us quite a lot of effort to put these together manually to ensure that they're correct when they go out. And actually, later on this week, there's a more specialized workshop to talk specifically about these biological quirks in birds. But we also get other quirks, like some insects may have very large volumes of repeat sequence, which gets assembled into very fragmented pieces, which we can put together manually, but it's very hard for the scaffolders to put together. So these are the kind of things that we're trying to work through in the manual curation process. So as I said earlier, our number one source of data to perform a manual curation is HiC data. It's really, really important. If we don't have HiC data, there's not a lot we can do. We just have to trust the assemblers. So for those of you that don't know what HiC is, I'm sure most of you do. I'm preaching to the choir. But it's a technique that allows us to essentially measure the frequency at which two pieces of DNA are found in close proximity to each other in situ, in the cell, in the 3D structure of the DNA in the cell. And that means that we can then use that data to rearrange the contigs of the assembly into the correct orientation that they're found biologically, and as a result, resolve the chromosomes themselves. So you can see, you can imagine that this frequency data of one-to-one matching between chunks of DNA can result, scale it up. You can result in a heat map when they're sort of plotted against each other, and that's why we have down the diagonal we have a really high intensity of signal, and then we have on the off diagonal we have varying levels of the signal. And this is indicating that, by the way, can people see my mouse? Yes, cool. We can see on the off diagonal signal, which is indicating that pieces should be moved around and rearranged to fit them onto the diagonal and resolve it. So you can see in this little graphic here, I'm going from raw unmanipulated to rearranging, and you can see that these pieces would be moved and flipped around to get the off diagonal signal onto the diagonal, which is ultimately where it should be. I've gone over that quite quickly. Jo will explain a little bit more about that when she goes through the map. So when we produce assemblies, they come out to varying levels of contiguity. So some assemblies come out and we map the HiC data and we produce these maps, and they're really clear, and there are hardly any misassemblies and we don't have to intervene very much. But equally, there are instances where the assembly has really struggled or the scaffolding has really struggled. So there's far greater number of scaffolds, and they haven't joined the chromosomes correctly. So we have to do that step manually. And this is another example of going on the left from pre-curation with lots of off-diagonal signal, manipulating it, in this case, in the software PretextView, and ultimately remapping all the HiC data to resolve chromosomes correctly. So we've gone from an n number of scaffolds on the left of 230. We've done all these joins and breaks and rendered curated assembly with just 62 scaffolds and the correct number of 9 chromosomes. One important thing that we often have our questions about from other institutes is the use of multi-mapping data. By default, this is sort of turned off by most users. However, over the years of developing Manual Curation, we've realized how important having multi-mapping read data is to be on. And this is a real prime example. So on the left, where multi-mapping is turned off, the HiC, you can see basically no HiC signal for all of these contigs or scaffolds in the bottom right-hand corner of the map. When we turn HiC mapping, multi-mapping on, you can see you do recover this signal. And it shows, although maybe you can't resolve the correct order and orientation, that they all belong in this mass of heterochromatin that joins two scaffolds. So they're two arms of a single chromosome that are joined by all these repetitive sequences in the middle, and that's backed up by this karyotype image, which shows this this chromosome with all this heterochromatin in the middle. I should mention now that our techniques are evolving almost daily, and recently this year we have been moving to curating 2 haplotype maps. So all of the ones previously that I've shown, they've been single primary assemblies, and we're just curating 1 haplotype. We're starting to move towards a system where we always curate 2 haplotypes. We're using the HiC phased data when possible from the latest HiFi assembler. And we're like I say, we're doing both haplotypes in 1 map, and then outputting a single haplotype as a sort of primary curated assembly. And what this allows us to do is spot any interesting inversions, differences between haplotypes. And also it really clearly indicates the sex chromosomes, because in these maps, in these dual haplotype maps, they are present in single copy, like you can see in this map on the right here. There are various other data resources that we use. So we will often do synteny analysis when there's a closely related species to check orientation or what should be joined together to make chromosomes. And likewise, we do the same with Busco painting and mapping between closely related species. All the tools that we use are publicly available. If not now, there are some tools that maybe aren't quite publicly available that we have developed in the past couple of months, but will be very shortly. But we're sort of in the process of updating all of our documentation. But most things are publicly available, and if not, you want to ask us about it, please feel free to get in contact with us. There's this Slack channel for support for assembly curation, manual curations. You can join this channel and speak to us directly on a daily basis. But this is the kind of general overview of a rapid curation process. We're starting with the sorry, just move this thing. Starting with the assembly fasta file, mapping the HiC reads, and creating a pretext map, which we then take into PretextView software, and we rearrange the maps, output our curated map, and then we have these couple of scripts to then render out our curated fasta file at the end. I'll put in brackets the S's in brackets here because we actually have the ability now to output multiple fastas for both haplotypes if desired. We can talk about that if you need. Generally speaking, when we're producing the curated genomes, we're outputting chromosome names ordered by size, but occasionally for well-established genomic resources, for example, like the 3-spine stickleback and the stickleback sort of genome community, we might produce a genome which we order and orientate with the existing reference genome. But generally speaking, we're naming chromosomes by size. And where possible, we identify sex chromosomes, but we only do this with caution. And we learn, predominantly speaking, we'll only do this for the heterochromatic sex, where we have one XY or ZW, for example. There are very few examples where we're happy to assign the homogametic sex chromosomes based on synteny. The examples would be Lepidoptera and so most mammals, but yeah, we do that with caution. for a single hap map, we can identify the heterochromatic sexes by mapping read coverage, and these ones come back as half coverage compared to the autosomes, so they're relatively easy to spot. Or like I said before, we're doing a dual hap map and they're present in single copy. So that's a brief overview of manual curation. Once again, we're the GRIT team at Sanger. This is the list of curators that are here present today who will be answering your questions if you have any. And again, there are some resources that can help if you have any problems. But really, the best thing to do, I would suggest, is either email us, grit@sanger.ac.uk, or join this Assembly Curation Slack channel, which we sort of monitor and can answer any questions. And there's a good community of other people that may also be able to help answer questions as well. So if you have any questions, as I said, please put those in the Discord channel. Someone will answer them. Now you're going to have the practical, where we're going to have a walkthrough from our expert curator, Joe Collins. In the Gitpod environment, there's various data available for you to have a go at. You can open PretextView and open maps. If you want to follow along what Joe's doing, she's just going to do a walkthrough on the screen. So you can either just watch her doing this, or you can open PretextView and follow along as she's doing each step. I believe we're going to start with this Il-5-Bati 1 curation. But as I said, if you have any questions, just pop them in the Discord channel. We also have breakout rooms available. So if you have a more challenging problem you want to ask about, you can ask to go into a breakout room. One of the curators can join you in there and you can share your screen and we can talk through what's going on. And just finally, this is how you are going to open up things in the Gitpod view. Run /PretextView and in the terminal to launch PretextView and allow pop-ups, and it will open in a new tab. And then you can find the data by clicking Load Map, navigate to Computer, Workspaces, Test Data, and then open Il-5-Bati, pre-curated pretext, and then you're good to go. So one last thing before I hand over to Joe is that to maintain the GUI activity and not allow it to crash, you need to type something into the terminal every 29 minutes. Otherwise, yeah, you'll be crying. So yeah, just
I will have a timer going that everyone should be able to see. So
James got a timer.
If there's annoying beeping going on in the background, like let's see if this works. So I'm going to start a 20-minute timer. Everyone should be able to see that.
Great, yeah, we can see it.
Cool. Right, so yeah, that'll go off in 20 minutes. So get cracking, I guess.
I should have had that for my talk. I've gone on for way too long. Cool. I'm going to stop sharing and hand over to Joe.
Thanks, Dom. Right, so hopefully everyone has managed to get their pretext map open. I'm just going to start sharing my screen now. Hopefully you see my pretext map. Is that what everybody's seeing? Yeah, cool. Okay, yeah, so as Dom mentioned, if you've got sort of quick questions as you go along, if you can pop them in the Discord channel, that would be great. And then hopefully the other curators can answer those for you as we go along. I am just going to do just sort of a very measured run through of how we do a curation within our system. As Dom mentioned, this is Pretext, and this is the software that we use to do our curations. This is actually a moth, and we do find that the Lepidoptera are some of our less challenging assemblies that we come across. They seem to assemble generally quite well. So we thought this would be a sensible curation to start with, just to do this walkthrough with. So what you can see at the moment is the assembly as it's come out of the automated assembly process. And each of these are scaffolds that have come out, and they have got, these aren't complete chromosomes at this stage. What we're looking to do is to generate a fully complete chromosomal assembly if we're able to with the data that we've got. This is quite a good example of a map in terms of it's not, although there's things to do, there's not loads and loads of what we call shrapnel, which are the smaller scaffolds that tend to congregate in this right-hand corner, where for whatever reason the assembly process hasn't been able to put those together. In terms of the quality of the HiC, we've also got very good contacts. For some of our assemblies, we have very poor data, which means it's quite difficult to be able to interpret that data because the signal isn't very strong. So when we're looking at the pretext map, I'm just going to sort of give you some instructions of sort of how to move around the map and what we need to do to edit. Apologies to people who already have used this, but for those who haven't, just hopefully it means that you can follow along, even if not all the way, you can at least have a play about with what you've got. So to move around the map, hopefully you saw that one of the prerequisites for this training course was to have a 3-button mouse. So for moving around the map, if we use our right mouse button, hold it down, we can navigate around the map, just sliding the mouse button around. That helps us navigate around. And then we use our scrolling button to zoom in and zoom out. And then we have, if we press U, we have a menu. Sorry, wipe that down. And this is where you loaded your map. But it also has other things. It has sort of certain edit modes and things that these are the things that are overlaid onto the map. So you can see certain things, context name enables. There are things that are on as default and there are things that you can add. Yours might already be set up differently. I have certain things I like. You can also, if you prefer to use your mouse buttons the other way, you can invert the mouse buttons, but I'll be using them with this button off. So if you want to follow what I'm doing, it's probably best to do the same. We have something in here already called the metadata tags. We will use these. I'll show you what we use these for later on during the process. These are default tags that are already set into the metadata tags, and we can also add other tags, but basically it's sex chromosomes, and so we can tag up our haplotigs and any unlocalized sequences that we might find. It's also possible, if we look under our color maps, to change your color of your map. So you can choose something that you prefer. I prefer to use something with a white background, so I tend to default to orange. I think this map actually comes up as one of the black background, but that's not a color that I prefer. It also shows you your edits. Once you start making edits, it gives you a list of those, and you can undo them. Waypoints is a way of just putting markers on your map to maybe, if you wanted to pinpoint regions of interest that you wanted to come back to, or highlight if you wanted to sort of discuss a region with somebody else, or that tends to be what we use them for. And your scaffolds, oh, it's not that one, sorry. Input sequences are all of your scaffolds that are within your map. And if you click on them, it should take you exactly to that scaffold if you're trying to navigate around the map. It also shows you if you break a scaffold, it will tell you that it's broken scaffold 1 into 1, 2, or 3 pieces, etc. And then as long as you're zoomed in to a certain probably to the highest magnification, it will take you to directly to that particular piece of the scaffold that you're looking for. Extensions, this is where we can add on our tracks that have been ingested into our map, and we use these. They're additional layers of evidence to help us make our curation decisions, help us make informed decisions. So generally we have the backbone coverage, we have gaps, we have these are gaps within the scaffolds, and then repeat density just gives an idea where we're looking at sort of highly repetitive region compared to the rest of the scaffold. And then we have our telomeres mapped on here as well. Just a side note, the telomere track can be a bit buggy in that it can move sometimes with the scaffold. So just be aware of that, something to be aware of. You have to switch these on in this version of Pretext. So you have to switch these on and off in the menu, but we'll leave them on for now. So I'm going to press U, and that takes the menu off. And if everybody's okay, we will start curation. Okay, it looks like everybody's just quickly looking at Discord. Everybody's okay. In this case, actually, I think we will start with, just going to bring my coverage up. If you go back down to extensions, you can move your plots. If you right click, on the box, it allows you to move and change color of your tracks that you've inserted. So sometimes, as you can see in this case, mine's gone off the bottom of the screen slightly. So I'm just going to move it up so that I can see it. So in this case, it looks like, as I said, this is Lepidoptera. They do assemble pretty well. There are some, what we're looking at for sex chromosomes, as Dom already mentioned, is that we use two different methods to identify them. One of those being coverage, if we've got the heterochromatic sex, and the other being a close relative if we've got the homochromatic sex. But as Dom also mentioned, we are being quite cautious now about which species we think that that's okay to do that with. And Lepidoptera are one that has, it has a well-established set, so we feel comfortable to do that. And in this case, it looks like, although we have some low coverage bits at this end of the map, which I will come to later, it looks like what we probably have here is the homochromatic sex. So because I'm worried about time, I would tend to do the sex chromosome stuff maybe later on in my curation, but I think I'm going to show you one of the methods that we use to identify the sex chromosome at this stage, just so that we can get that kind of done while everybody's still online. So I opened this earlier just in case it didn't work. So I'm going to just refer you back now to your workspace, and at the top you will see this DFTB PNG, and if you double click on it, I think that's how I got it to open. Somebody correct me if that's not right. What should open is this Circus Busco-Synteny plot. And this is where basically, I mean, we have a script that we can use in-house to identify a close comparator, or you can go in via the NCBI taxonomy browser, and you can, take your specimen and find something that's a close relative, and then we would run an alignment with the, what you've taken to identify, in this case, our Z chromosome. And what we can see from this circus plot is that, so this is our reference, and this is our whatever it's called, Thylarobatis assembly. And what we can see is unquestionably a really close match with this is our reference Z, and this is our scaffold 1. So without any doubt, we can, providing that we're using this and we're comfortable to use this as a good comparator, we can come back to our pretext. We can come back to our pretext. Sorry. And we can label up our Z chromosome. So the way that we would do that is we go back to, sorry, on our keyboard, if we press M, that brings up our metadata tag menu. Again, it's slightly going off the screen for me. And if we use our, sorry, just because it's off the screen for me. Sorry, bear with me. I'm just going to move out the screen so I can see it. Right? Sorry, it's putting tags all over the place. Has anyone else got the same issue as me with it being off the screen? Sorry, everybody. 1 sec. Get rid of that, right. Pressing M again. It's not showing me. I'm going to show you what you should be seeing. Okay, so what you should see, so this version I've got open separately. So if you go to your M menu, your metadata tag menu, what you should see is if you scroll left to right, you can see that the active tag in the metadata tag menu changes. And what you want to do is you want to use the left and right buttons to find the Z, and then using your left mouse button, you want to click on that scaffold and then place a Z on it, because we know that that's the Z chromosome. Right, let me just return back to this. Has everybody else, can you all see the whole metadata tag menu? Curators, sorry. Or are you having the same issue as I am?
I can see it all. It might just be you.
Brilliant. Okay, in which case I might go back to my, I just can't, it won't move up there. I'm going to go back then to my other version, I think. Okay, which is the same, it's just not in the, I just hope everyone else at home can use that. Okay, right, I will carry on from there. Okay, so we've, in this instance, we've used the Briscoe synteny plot. We've labelled our sex chromosome because we're comfortable that the evidence that we have for that is sound. And the other thing that we would do with Lepidoptera, when we do our submissions now, we make a note of the species that we've aligned to in our tickets that we use in our submissions. Just so that should somebody need to come back to that, there's a reference as to why we've named our sex chromosome. Right, so what we're going to do curation wise then, so we've done moving about the map. Now we're going to have a look. If we press E, we have our edit menu, and you'll see that when you press that, you get this crosshairs come up. And that's what allows you to pick scaffolds up and move them around the map. So this is what we're going to need to do in order to curate the map fully. The other thing that allows you to do while you've got something picked up, so you press the middle button to pick up a scaffold. Sorry, I forgot to say that. The other thing that you can do whilst you've got hold of the scaffold is that you can press your middle mouse button and that inverts the scaffold. You can see from the arrow that it's inverted, and then if you put it back, the arrows come off. To put the scaffold down, you press the left mouse button and that places it down. As soon as you've finished editing, you want to press E to turn the edit menu off, because it's really easy to accidentally start moving things about and cutting things up. When we edit, we tend to have, I think it's default, it comes on with this snap mode on. So to turn that, it comes up with that on, I think, or unless I've got that on, but that's S. When you've got the edit menu on, that will be S. So to switch it on is S, and then to switch it off is also S. The time that you want to switch it off is that if you've got a small piece in your shrapnel that belongs within your scaffold and that wasn't placed in there as part of the automated process, you would need to press S in order to put your small piece of scaffold within the larger piece. So if I just show you now, so with this is with S on, I can move my scaffold within the other scaffolds. And now if I turn S off, with S off, that I can move my scaffold within the other scaffolds. And now if I put S on, I'm not able to do that. I can only go between scaffolds. So the reason that we want to do that is we want to do most of our editing with snap mode on, because it's very easy otherwise to insert or to make small pieces accidentally. And this can upset the sort of upstream process when you're making your TPFs, because the scripts can see small pieces that you haven't, it's not done by design that you've made, you've just done accidental things, and that can confuse the script. So that's not ideal. Right, so what we'll do now is we'll start to go through the map and try and put this genome together. So as I mentioned before, we've got all of our primary scaffolds on the diagonal. We've got this load of smaller scaffolds at the end, and what you'll be able to see is that we're looking again, we're looking at the small scaffolds against the scaffolds, the primary scaffolds on the diagonal. And what you can see is that in some instances, some of these small scaffolds have really strong affinity to some of these larger scaffolds. So things like this one would belong with this one, and this one would belong with this one. And that's really what you're looking for in terms of pulling scaffolds together. The other important thing that you're looking for when you're looking at the scaffolds that have already been made is that you're looking for contiguity and a really good strong signal on the diagonal to help determine that your scaffold that the automated process has provided for you is actually assembled correctly. And often it's only once you've looked through those scaffolds that are sort of the primary units that are already constructed, and you've looked to see whether they are assembled correctly. Sometimes it's only once you've done that that you can actually properly insert the small scaffolds that should be part of your chromosomes. But that depends. Sometimes you can insert them, easily if it's just a case of a simple gap. Other times it needs a bit more work in sorting out the assembly before you can construct the chromosome as it should be. So the way that I would start a curation is starting in the top left corner. We're looking all the time to see this very strong signal on the diagonal, this very dark signal. And that's what we're looking for. We're sort of going down the map, just moving. I'm using my right mouse button, just pushing down, holding that mouse button down and just sliding along just to have a look. So that to me looks like a pretty good assembly. If I wanted to, I could press my U button and turn my menu on and put my gaps on, and I can see that there are no gaps in that scaffold either. So it's likely that that's a well-assembled scaffold. The other thing I can do is I've already got my telomeres on. So if I scroll over my telomeres, I can see that there's a telomere on this scaffold as well. So we often find with the Lepidoptera, the telomeres, they do tend to be T to T. Quite often that's just how they assemble. So now we've looked at the large scaffold, we want to see if there's anything now in the shrapnel that needs to be added to that scaffold. And sure enough, we come over here and we find there's one piece of shrapnel that has really, really good, I can't think of the word, really, really good affinity, sorry, to the large scaffold. So what we do is we can bring that over, and I'm going to make sure that I press my S now. So I'm press S and just put it next to the scaffold that it belongs to, just so that we can just talk about what we're looking at here. So we can see that this small scaffold undoubtedly goes with this large scaffold because it has really strong affinity with this scaffold and none of the other large scaffolds. So we're absolutely certain it goes with this large scaffold. The other thing that we can see really quickly is that the affinity is strongest at the top. So we know instantly, so sorry, I'm going to pick that up. I'm going to middle click to pick it up and then left click to drop it. And I'm going to press E to turn the editing function off so I don't do something horrible. So I've just placed it there. So these coloured bars at the top, I should have said before, these are what we call scaffold identifiers, and they just help us see, very quickly if we're looking at one scaffold or parts of one scaffold or a separate scaffold. So if you press I on your keyboard, you can very quickly toggle them on and off, which I find quite useful sometimes because sometimes it's covering a signal that that I just like to be able to see clearly. So I've placed this, by HiC affinity, I've placed it definitely at the right end of my chromosome. So now we want to make sure that we've got this continuation of this affinity, this diagonal all the way through our chromosome. And if we look where we've placed this, we can see that actually we've got a gap in affinity at this point. And if we look at this end versus this end, we have much stronger affinity between these 2 points. So that tells us really clearly that what we need to do is we need to invert this scaffold. So at this point, we'll put our editing menu on again. We've got snap on already on, so that's great. We're not going to create any nasty little small pieces. I'm going to use the middle mouse button to pick it up. And then I'm going to press the middle mouse button again, and that flips it. So now we can see that the affinity between the scaffolds is much stronger. So to drop the scaffold, I'm going to press my left mouse button and I'm going to press E, and that takes the editing function off. So now we've got really good strong affinity between our scaffolds. And if I press I to take my identified tags off and G to take the grid off, you can see now that we've got a really nice strong affinity all the way through that region. So that tells us that what we've done is we've correctly assembled that small piece of shrapnel as part of that scaffold. Okay, so I think that was the only piece of shrapnel for that. Yeah, so we're looking now to see if there's any other strong scaffolds with strong affinity. What we're seeing here at the bottom, these are sort of telomeres. This very strong signal is for the telomeres. And what we see often is that you'll get that at the ends of the scaffolds and you'll get that at the end of the shrapnel scaffolds that have telomere on themselves. But what you're looking for, in this case, you can see that actually over the length of the scaffold, or chromosome in this case, you actually don't have much affinity. So that just tells you that actually, although it's matching strongly at the telomere, because they're sharing telomere repeat, these shouldn't be part of the scaffold that we've just worked on. So now we're going to come down the map to our next scaffold. So this is telomere on the end. And we're going to just using my scrolling button just to zoom in. And then I'm going to use my right button just to go down the map. And we're looking for that strong contact. And actually, in this case, we've got strong contact all the way through. That's this end of the map. It's not that one, sorry. Okay, right. Sorry, let's go. So now what we're going to do is we're going to do the same thing. We're going to go back to this end and we're going to look for things that might want to go within our scaffold and be part of that chromosome. So unfortunately, we have to take these one at a time because the software doesn't allow us to sort of pick up multiple pieces at once at the moment. So again, we're going to press E for edit. We're going to use our middle mouse button and we're going to drag our piece just by moving our mouse. We're just going to drag our small piece of shrapnel over to our large piece. And I'm going to left click and press E to put it down. Sorry, I don't think I did have snap mode yet. Make sure you've got snap mode on before you do that. And what you can see already is that actually the correct position for this scaffold is for this piece of shrapnel actually is at the end of this scaffold, because we've placed this, we have strong contact all the way through that region. You can see as well we've got a bit of telomere on the end because like I mentioned before, in Lepidoptera often the scaffolds come out of the auto-assembly process T to T, and not only can you often see that on the telomere track because it's been tagged, the telomere repeat motif has been tagged, but often you can see it visually because we can sort of look at regions on the ends of our chromosomes that sort of light each other up and have that sort of cross affinity, and that also helps us determine not just in Lepidoptera, but in other species where they can be more difficult to assemble. That's quite often a good tell to see if you've got your assembly arms constructed in the right order and orientation. Right, so there was another piece of this scaffold. So we're going to go back and we're going to get this piece. So pick up, I'm going to use E again, middle click, pick up the scaffolds. And then I'm going to do what I did before. I'm just going to leave that at the end for the moment. And what I can see is actually the strongest point of contact for this scaffold is in the middle of this chromosome. So I'm looking for this dark region here, and I'm looking at that versus the rest of the chromosome. And what I can also see, because I've got my gap track on, is that there's a gap at this location. Now that is fairly typical, and it gives you it sort of gives you a good spot to drop your scaffold into as well. Makes it easier for you because that's typically what we see is these scaffolds end up going into the gap. So this time, sorry, let me start that again. This time, because as I mentioned before, with snap mode on, you can't move your shrapnel scaffolds within other scaffolds. So this time, in order to place this piece of shrapnel within the larger scaffold, we need to press S on our keyboard to remove snap-on mode, and we need to drag our scaffold within our primary scaffold. So what we want to do is we want to place this pretty much sort of directly on the gap, usually just by eye, sort of looking where the best place is to put it. So once we've got it in position where it looks like the signal's looking correct, and in this case we can see clearly that this is the right way up, because we're looking at this signal here where it's a nice strong signal all the way through the join, and this signal here where it's also a nice strong signal. If we were to invert this, I'll just show you what it looks like. You can see that it looks wrong, the signal's off. So sometimes it's a bit unsure, but if you just sort of have a play with the scaffold, you'll usually find that one way up is definitely preferable than the other. So in that case, we've put that small piece in. Okay, let's go back, see if there's anything else. No. Okay. So, right, okay, so we're going to do the same now with the next scaffold along. We're going to go all the way through, looking really carefully through our diagonal just to check that what we're looking
Joe,
Yeah.
now might be a good time to get people to go into their Git pods and have a click around.
Oh, sorry, I thought that's what they were doing.
Oh, into the
Oh, yeah, I'm with you.
yeah, the terminal side of things.
Okay, just if everyone wants to stop and
Maybe a quick hello world or click through a few PNGs, and that should wake it up enough for the next 20 minutes.
Assuming a quick LS is,
yeah,
Should be fine, yeah.
or something. Okay, hopefully everybody's done that. Okay, I'm going to also have to go back to my map. Okay, I'm going to carry on now. So hopefully everyone, thank you, Damon, for doing that. I'll carry on. Give us a shout when I need to start next. Right. Okay. So that was. In the order. Okay. So then I'm going to come to my next scaffold. I'm going to do the same thing. I'm going to look through my diagonal. Got no gaps in here again. It all looks good. I've got strong contact all the way through the diagonal, so I'm happy with the assembly that it is. This is what I was talking about with the telomeres lighting each other up, and also the lighting up neighbouring telomeres, which is always a good sign. And then we're going to go across again to the shrapnel at the end and see if there's anything that wants to be part of this scaffold. No, it looks okay. In this case, it looks like what we've got, we're happy with. So we'll go down to the next one and repeat the process. So that again, all looks good. Good contact all the way through. Right, so in this case, it looks like we've got two small scaffolds that want to join. And so I'll use E again, put my edit window on, middle click. Whilst I've got that piece attached, I will scroll using my right mouse button over, and I'm going to place that with snap mode on the top of my scaffold. Oh. I'm going to go back and get the other piece, which now you can see, because we've moved one piece in, now helps us determine the order of both of them. So it looks like this piece goes on the end. So now we've put two pieces of shrapnel in, place those next to each other. Won't do that now. So we used one piece to help us determine the order of the other piece. So do I through here. Now we've got contact through those pieces onto our assembly, onto the rest of the scaffold, sorry. We've looked through that already so that we know that we're happy. I think that was all the pieces that we had. Yeah, so it looks to me like that was everything. Yeah. So we're just going to keep going down the map, doing the same thing, trying to assemble these scaffolds into full chromosomes. So in this instance, we've got good contact all the way through our diagonal to this point, and then we've got some white space. So sometimes you see this when you have sort of quite repetitive regions, quite densely repetitive regions, and often that's just part of the sort of HiC motif that we can see. But sometimes it's a case of that portion here, which has white space, doesn't have any contact with the rest of the assembly, might belong elsewhere. So it's always worth going from this point and having a look at the rest of your map to see if actually that piece belongs elsewhere. And actually in this case it does. It looks like that small piece needs to come out and be part of another scaffold. It's part of another chromosome. So I think in this case, it's just, so potentially I'm guessing these are all bits of the same chromosome. Yeah. So what we can do is it's quite often that you find these pieces, they, like other regions where you have missing data or if you have misassembly, quite often these pieces are inserted next to a gap. So in this case, we will press E, and then what we need to do is we need to use our crosshairs to basically capture the region that we want to move. So as accurately as we can by, if you basically to widen or narrow the distance between these green blocks, you sort of move your mouse, slide your mouse up and down on your worktop, and that helps you get your crosshairs to be the right shape, the right distance apart to capture the piece that you want. So I'm just going to now say, best if you can to zoom in as far as possible using your scrolling button, and then press E, and then going to go as close to the gap as we can at one side, and then just going to by eye using, what I tend to do is look at where the green haze is on the white, where it swaps at, you know, where it's just contained in the white region, and then we will just grab that piece by, we'll grab that piece by clicking L, and then we have to press S off to get it to move out, and then we've picked up that piece, and then we press, to wait till we come out, and then you can scroll over as you have been doing using your right mouse button to scroll. We'll scroll over to the right chromosome, and it looks like it's part of here. Now it's not possible for me at this stage whether there's something else that goes in there, and I can't really be sure where that piece of sequence goes in. It looks like it goes in around there somewhere, or it could be, just going to look here and see, I'm going to look at my coverage to see if there's any indication that that might be a haplotig. It's not clear. So you can play about with your scale for your coverage. The default coverage is quite a I can't remember the word, but I tend to lower the scale on my coverage just to make it easier for me to see, to determine if something's haplotic. I can't quite determine that at this stage, so I might help me later. I think there's another haplotic that goes in here, so possibly it is. I'm just going to leave that there later. And this might be where I would use a waymarker tag, just to know that I need to come back to this to get some confirmation. So in order to use a waymarker tag, we press W, and then you can left click on that piece. So that puts you a tag on so that you know that that's something for you to come back to. So what we'll do is we'll go back to the scaffold that we took it from, which was scaffold 5, and we'll carry on from where we were. So we've taken out that piece of chromosome that doesn't belong there, and we're now going back through the rest of the chromosome to make sure that we're happy with the assembly that we've got. So that all looks okay. We've got telomere on the end. We don't appear to have anything else that goes in. So we will continue the process. We are going through the scaffold. Let's see anything else, and we will go again to look at the shrapnel. And again, there's nothing with a strong contact that needs to go in there. Okay, come back. And then we will go through this scaffold. Same thing again. Scaffold, fidelity looks good. Nothing in the shrapnel to go in. So this is quite a quick curation so far. Again, we're looking through, looking to see that contact. All looks good. I think there's a tiny piece there actually. I've got a piece that goes in there. So that looks like, looks like it goes on the very end, looking at its contact. We've got a couple of pieces there actually, one we can't tell. So in terms of how much of this shrapnel you want to include, I think we would all say that it really depends on the genome. Sometimes you end up with loads and loads of tiny pieces, which is, you know, because of the time pressure, you can't go through all of those pieces. It's not practical to do so. In a case like this, where the assembly is pretty good and you've only got a few pieces really to go through and it doesn't really take a lot of time to do it, then it's, you know, it's feasible to do it. In other assemblies, which with very large genomes, you have to be a bit wary because some of what looks like quite small small shrapnel is actually quite large chunks, which, you know, which they could have a lot of gene information on them. So it's kind of a sort of almost a case-by-case situation where you're looking at the shrapnel. If it's repetitive shrapnel that it can't really be placed, then obviously, you know, you don't want to invest too much time trying to get that sorted. You might be able to localize it to a chromosome, but and sometimes you can find a location within that chromosome. But often we find that with repeats, we see mappings to multiple chromosomes, or it's not possible to get a proper handle on where those shrapnel scaffolds locate within that chromosome. So it's best not to spend too long dealing with those. But in this case, you know, it clearly goes on the end, so we can usually roll out, take our small shrapnel piece over. We've got snap mode on, looks to me. No, I think it does go the other way. And I think it's the right way up. So we just add that to the end of our chromosome. I think there was another small piece here, but it was tiny. And I think it would be difficult to, you know, just to say where that goes in. I can have a look at it, but I don't think I will spend too much time bothering with that. Best case, you'd probably want to unlock it, but I think most of us probably wouldn't bother in that sort of instance. So sorry, reason I said that so that I could show you. So what we have on the metadata tag menu is there are, like I say, these regions that sometimes we can localize to a chromosome, but we can't actually determine where on that chromosome it goes. So in that case, we can, on our metadata tag menu, we have an option which, if we scroll through using the arrows, we have an unlock tag, and we just using our left mouse button, we can put a tag on there, just so that when the assembly gets processed, that unlock fragment will be named an unlock for the parent fragment, which means they will go through as part of the same chromosome. So that just keeps, you know, it's a way of sort of saying it's you know, it's a way of saying that this data, we know it belongs here, but we can't place it, but it still might be useful information for you to know that it's part of this scaffold, that part of this chromosome. Right, sorry, I'm trying to do this quite quickly. So we'll go on to the next one, going through. And we're going to look here, and again, we've got another piece here. So pick that up and drag it across. And again, and park it to the right of the chromosome. In this case, I'll turn my gaps back on. You can see again that the strongest contact here is where this gap is. So we're going to maneuver it into the right location. And if you can see again that the strongest match, although there are it's not quite as clear as in the other one because there is a match. There's a sort of a high affinity on both ends of this chromosome. So, but this actually has the strongest match. If I flip it the other way, you can see that actually it looked better before. So that tells us that our piece is assembled correctly within the chromosome. We'll go back. That's that one. Sorry, I'm going through this quite fast so that we can get to the next phase. So again, just looking for that really strong contact. Nothing in the shrapnel. Looking for the contact. All good. We did have something here. Again, we've got a small piece, so press E, middle click, scroll out, drag the piece across, making sure snap mode is on, left click to drop the piece. The contact tells us where to place the piece. We've got really strong contact between the left end and the right end of the small piece of shrapnel. So it's great. Right. So now I'm just going to take my gaps off for a second. So what we're looking at now is, so we're starting again as we normally do in the top left-hand corner. We're looking at our strong contact on our diagonal. And what you'll see here is actually there's a loss of contact through this point. What we can also see if we put our gaps back on is that there's a gap here. And the other thing to note is, as I mentioned before, particularly in Lepidoptera, the telomeres that we see often light each other up. They often have affinity with each other. And we've got telomere on the end here, which we know we're happy with the assembly of this chromosome. And what we're seeing is actually that this telomere here has affinity with this region in this assembled scaffold. So taking all the evidence into consideration, we've got a gap here, we've got loss of signal here, we've got telomere sequence here, affinity between telomeres here, and we also have actually, if we look here, we've got better affinity with the end of this scaffold and this end than we have at the moment. So what we're going to do is we're going to invert this piece of sequence here. So we're going to use E. I'm going to put my gaps back on, sorry, so that gives us a guide of where to cut. And we're going to use E again. And remember, we put E on, we've got snap mode on. We're going to drag our mouse button so that our little green blocks are in roughly the right location to cut out the piece that we're interested in changing. Then we're going to left click to pick up just that piece. Then we're going to middle click in order to invert that piece. And then we're going to left click again to put that piece down. And now if we take a look at what we've now got telomere sequences lighting each other up, and we have affinity through the region which previous to this had lost affinity at this point. So we can be fairly confident that we've made a change and the change has now corrected the assembly over that region. So we'll go back through the rest of the chromosome, which all seems to look okay. We'll have a look. Right, so now we get to the other part that I mentioned earlier concerning coverage. So we can see here that we have really strong affinity between this quite large piece of scaffold and the chromosome we're looking at. So I'm just going to use E to pick that up. And we're going to place that next to the scaffold that we have already. So it looks as though this, based on HiC affinity, this piece of scaffold should be part of this chromosome. And our assemblies are haploid, and basically this is the other haplotype. So what we're looking at here is haplotypic duplication. The reason that we know that is because before I was mentioning, I think Don mentioned as well, that sometimes when we use our coverage track to help us identify the sex chromosomes if we've got the heterochromatic sex. But the other thing that we use our coverage track for is to identify regions of haplotypic duplications. And basically what we can see here over this region is that we have our coverage track, which is choppy anyway, which we expect, but it's a fairly steady threshold of coverage all the way through our autosomal regions. And then what we see is this massive drop of coverage, and this drop to half coverage occurs exactly the point where we also see this duplication occurring. And what that basically means is that instead of the autoassembly process putting just one haplotype in this haploid assembly, what we've got is a chunk of the other haplotype as well, where there has been some sort of confusion from the assembler. Instead of booting out one copy, it's kept both copies of this region. So it's up to us to manually remove those regions because we don't want false duplications of part of our assemblies over the same region. So what we need to do is we can see that the scaffold we've got is perfectly well assembled. So we don't want to make any changes here, but we want to make sure this is removed. So if we use it's probably easier actually to see. So basically this shrapnel scaffold is the same as this piece of sequence. So what we will do is we will use our metadata tag menu again. And if we scroll through using our left and right arrows, we can find a tag that says haplotig. And if we place the haplotig tag on there, what that means is when the assembly goes through the next stage, through the processing stage, after we've done the manipulations that we need to make, that the scripts know to remove that scaffold from the next phase. So that will not be part of the final curation. I don't think that's me. So cool. Okay, right. So hopefully, yeah, that explains about the haplotigs. So yeah, basically they want to be removed. Right. So that's that one. Let's come back down. We're looking through again. Through, that looks okay. Looks okay. In. Looks okay as well. You can put in. We'll come down to this one. So if this looks okay, let's maybe let's get it there. Oh, this was our piece where we moved this little piece to. This was our chromosome where we moved the piece 2 that we took out of the other chromosome, and we just left it there with our waymarker tag on. So we can see already this is looking very familiar. We've already looked at one of these. So let's pick this piece up because we need to remove it anyway. So you can see exactly the same issue as before. So we've got the really strong, clear diagonal line showing us the part of the chromosome that's duplicated. And if you look within the chromosome where that duplication is, we can see exactly as we saw before that the coverage over that location is halved, and that matches the half coverage of that entire scaffold. So we need to tag that scaffold up as hapdupe. And what we can see, I'm going to take my waymarker tag off now. We do that by pressing W, and you have to middle click on the little horizontal line in order to take it off, because actually it looks like, when I look at the coverage, this small piece has similar coverage to this region, and it just looks like it goes on the end here. So I'm going to put haplotigs on both, haplotig tags on both of those regions. So they're tagged up and they will be removed. Let's come back here. Looking at these already, I can see both of these pieces of scaffold want to join this chromosome, but actually they both want to join in the same location. And one of them, they have sort of, looking at their HiC affinity, they have slightly differing contacts. So it's likely that these may be haplotypes of each other. So we could have taken those at once, actually. I'll do that so you can see you can do that. So you can take 2 pieces. If you want to move 2 pieces that at once, you can do that as long as they're next to each other. So if I press E and I click my middle mouse button, it will pick up one as we've done before. But if I hold down that middle mouse button and I drag my cursor over to the next piece and then let go, it picks up both scaffolds at once. I'm going to move them over here And once I've moved them, you can see what I was talking about. So actually these two pieces, we could tell because they both wanted to go at the same end of our primary scaffold. These two pieces are actually haplotigs of each other. So we only want to keep one of those. So that one has a stronger match. It doesn't really matter which one of those. I've just kept the longest one because it's the same sequence. Sometimes it's possible to tell. Some of the assemblies that we look at, there's a really strong sort of haplotype bias. You can sort of piece together things by haplotype. Where the autoassembler hasn't done such a good job, it's possible to sort of manually phase assemblies. But in this case, I've just taken the longest copy. And again, you can see the coverage on here is another indicator that this is, you know, it just gives you good evidence for showing that it's the haplotype. I think that's all of that.
This is your second 20-minute timer.
Okay, try and write something. Thank you.
I'll put an alarm on the next one so everyone can hear it at the same time.
Jump. Oh, I'm not working that. Hang on a second. Right, okay, so I'm going to try and get through this. I'm just going to carry on and go through this, try and go through quite quickly so that we can get to the next bit. So again, I think I'm going to take this piece first because I can see that this piece I can see as part of my chromosome, but I think I need this piece first because that has the strongest match. So put it to the right end, I flip it to make sure I've got the strongest match there. I might have something on the end that doesn't belong there, maybe. And then I go back. And now I've got the strong contact with the end piece. So I had to put one piece there and then it gave me the ability to see what I needed to put next. There's a tiny piece, yeah. Okay, so on the end here, if you can see, I'm going to remove my identifying tags. And you can just see there's a tiny piece here with lots of white space, which is a similar thing that we had before. And actually, if you go up, I should have remembered which one it was. I'm just going to see where it was now. Actually, right, because I've confused myself as to where it is. Right. If you want to go backwards on pretext, if you want to undo something, I'm going to do this now because it's a good time because I can't remember where I saw that this matched. I've just put that there, but what you can do is if you press E and then press W, sorry, you need to be, no, it's not going to do it now. Sorry, Q, it will put, so if I just move that out and then press Q, that puts it back where it was. So you can undo what you've done. And if I want to take it out again, I press W and that removes it again. I'm just going to put it back and then I'm just going to remember where it was that it needs to go because I forgot. So it needs to go as part of level 17. Okay. I'm going to use E, use my crosshairs to guide me where to take it. And I'm going to drag it up the map. Hopefully remember where it needs to go this time. Here we are. And then it looks like it wants to go in at the end of this scaffold 30. That's a haplotig. Okay, so I'm looking at the coverage. Yeah, we can see that it actually it's likely, sorry, it's likely part of this large haplotig scaffold. So again, we're just going to tap that with a haplotig tag. So right, we'll put those pieces on there. Okay, we can keep going. Anything there? Sorry, I'm going through quite quickly because I want to get to the next stage. Nothing there. Nothing. Well, it's a bit less of a signal, but we've still got signal, so, and there's no gaps there, so it all looks okay. We can see we've got this large chunk that wants to be part of this scaffold, so we're just going to move that in. It's a bit lacking in signal there. Happily, we've got something that goes in between those bits that we've moved in, because that helps us. It's just another piece that fits in there, and that's actually got signal on that we need. It's not great, but I think it must be like that because it has better signal with the scaffold that it joins into. I think that was everything. Okay, and let's go down to the next stage. It's okay. We've got another scaffold here with a really strong contact. So we'll pick that up, move that in. We've got a nice contact all the way through our join. Now we come back, we've got another scaffold, really strong signal. The bottom scaffold, now we've got really strong signal through that join. Look through here, nothing obvious to break. Again, we've got a piece that goes in, we've got a very strong signal at the end of this scaffold. We move that in, we can see straight away that we need to put that back on, straight away that we need to flip this to make our join. I'm sorry, I should just go back and check, that's that. And then through here, looks okay, it's 2 there, that's okay. And so it looks like we've got some data to put in here potentially. It's like it goes like that, so in the middle. And just grab the other piece. Few bits to put in here, so I'm just going to bring them over to see. That will help us. Let's take my I'm just looking to see if that small piece that I've just brought over belongs elsewhere. I think it does. A strong match with that. So I'm not sure I'm going to leave that unsigned in the assembly because I'm not entirely sure that is I mean, it looks like it is part of this, but it's not overwhelmingly so I'm just going to leave it there for the moment. Right, so carry on going through. That looks okay. You've got a really strong match here. We'll bring that over, confident with our join. Again, this piece, so I've got my S off. I'm just going to put my, I'll leave it there for a second. So I'm going to put my gaps on. And as is typical, we have a gap at this location, and we can flip and place our scaffold in. That's everything for that one. Okay, we're all okay there. We've got a couple of scaffolds we can bring over together. So that one strongly matches with that. That one seems to be coming through there as well. And look, we've got another haplotig. And we can see the drop in coverage over the haplotig. So we'll use our our metadata tag mode, to put our haplotig tag on. We'll look through the next one. I'm just going to very quickly do the rest of this map. I think we're nearly there. Again, I'm looking at this now, looking to see that we've got a gap in this, a gap in our contact, and we've got a physical gap as well. So it looks to me like what we probably need to do is to flip our end. And then we go back through the rest of the scaffold to have a look. And then actually, to give us even more confidence, we have a scaffold that now wants to sit in the middle. We invert it. So now we've got contact all the way through. So that helps us confirm our edit that we made. This is another scaffold that I can't determine whether that wants to go in or not. That does have quite strong coverage. So I can't see exactly where that goes. So that might be an instance again where I use my menu. I'm going to unlock, tag that with an unlock. I think that's all for that one. And then we go through here. It looks okay. Signal's a bit low there, but not low enough to be overly concerned. You can't see it really clearly, but there is a strong match right at the top corner. So just going to flip that. And now we've got our good join. They're a bit not strong enough evidence really. So we're going to look through here again. It's good evidence. Good evidence for contiguity. Strong contact all the way through here. Again, we're looking here. We've got another strong contact at our top left. So now we've got good contact through that region. We've got a small piece as well, but could go in the gap. We will just put our other piece. Actually looks a bit like a hat based on coverage. I'm going to take that out and tag that. Using my menu M and then left click to put, oh, yeah, that's another thing to be careful of. It stays on the tag that you last used. So to remove a tag, you middle click, but you can only remove the tag that you've put on if it's on that position in the menu. So if I put unlock on there and I move to tag X, I can't use middle click at that point. It won't remove the tag. So I need to go back to where it says unlock, then remove the middle click. And actually what I want is to left click. Let's want to go to the haplotig menu and then left click to put hap on there. Right, and now we're at the last one. What we can see here is we've got two scaffolds that go together. We can see really clearly the duplication that's in the middle of the chromosomes, just in the middle of the join. So what we're going to do is we haven't got gaps here, so we're going to have to use our, just by eye, we're going to have to use our crosshairs, and we're going to sort of try and put our crosshairs in a position where we get all of the duplication without removing too much. And what we want to see once we move it, so I'm going to put S on so I don't accidentally put that, make a small scaffold. What we want to see is that I have made a small scaffold, I'm still doing that typically. Oh, that's really helpful. So I will do that again and remove that. And I'm going to tag that as haplotig. And now we've got contact through our join where we want it, but we've removed the haplotig, and we can see from the coverage that that is the haplotig that we want to remove. Right, so for the next stage, I am going to, so now that's pretty much the curation done. And what we're going to do, we've labelled our sex chromosome, we've curated our assembly, and what we want to do now is to output our assembly, our curated assembly. So the first thing we need to do is it's on the menu, you can have, I've already painted this, but I'm just going to remove that so I can do it again. Right, under the menu, there's a button called Scaffolds Always Visible. I don't know if is that default on? I'm not sure if it is or not, but that's what you want to have on. You want to click that on. And there's two ways of doing this. But basically, we're going to paint the scaffolds up so that when we do our output, that's going to represent the assembly as it is now, as we've edited it. So there's two ways of doing this. So if you press S, the edit mode menu comes up. And you can either press, you need to left click, and if you left click and hold your mouse down, it will paint the region which you're scrolling over. So that's one way to do it. And then you go one at a time down the map. And you have to sort of go through the diagonal of each of the small pieces, making sure that you pick up all of those tiny bits. But that's because you might have assemblies where you have got lots of small bits. Actually, that's not the easiest way to do it. So I'm just going to remove the tags that I've done. So I'm now going to come to the other end of the menu. And the way I do this is called flood fill. And basically that means rather than trying to pick up all the tiny pieces to paint yourself, you can use the tool that it has, this flood fill tool that it has, in order to sort of make that easier for you. So how I always start this is I click on a piece of shrapnel that I'm not going to include. And then what you want to do is you want to, holding A down on your keyboard with the painting menu, with scaffold edit menu open, you hold A down on your keyboard and then go to the top left region of your chromosome, hold A down and then left click. And that paints everything to the right of your where you've clicked. So I'm just going to unclick that as the way that I've always done, just find it easier. It's fine if you include anything you've tagged as haplotig within that, because the script at the next stage knows to remove it. So again, I'm going to hold A down and go to the top left. I always like to zoom in to make sure, because sometimes if you put in a tiny scaffold at the beginning of your chromosome, it's really easy to accidentally not pick that up. So it's always useful to zoom right into the top left-hand corner. Right, and this is where I want to mention unlocks. Unlocks have to be painted as part of your chromosome. And the other thing that's important to mention about them is that they have to be right next to the chromosome. You can't have, say, a small scaffold which you've tagged as haplotig in between the unlock and the primary scaffold. The unlocks have to come directly after the primary scaffold, because otherwise the next part of the process gets confused and it calls the tiny unlocks separate chromosomes, and that's not a scenario that you want to have. Right, so I'm going to carry on going through using A, just zooming in. And zooming in. As I know, I put some small pieces on the ends of some of these. An example there. So it's very easy, I think, as we can all contest to slightly mispaint. It's another example there. And then it can lead to a whole host of irritating problems that are sort of unnecessary. And there. And I'm just going to take S off because it's really easy to start accidentally painting things as well. So now everything's painted and labeled up, and what we want to do is we want to make our AGP. So if we press U on our menu, you can then generate your AGP, and then I can't remember where do these need to be, Joe? Are they in? Disappeared. Does anybody remember where the location is for worktop or desktop?
It should be, for the attendees, it will be computer workspace test data.
Because I can't open, so somebody else might have to do the next bit, because my as you noticed, my Google Chrome has died. Joe, can you do the next bit? Sorry, because my
Sure.
Git died.
Sure. Mine should be still working. Hold on, let me share my screen then. If you stop sharing, I can.
Yeah, sorry. You were going to start pretty much now anyway, so hopefully it's not too bad.
Okay. I'm assuming people can see my browser window and see a painted resolved map of our LEP assembly. Okay, so if you press U, you can bring open the menu, and we want to click on Generate AGP. And if we go to Computer and scroll down to Workspace, and we can place it in the test data, and we can save this. So override will overwrite the file if you've already saved it. So be careful here if you're trying to work on two versions of for some reason. Format singletons, this is important if you are changing the orientation of a single scaffold piece. So by default, we would normally check that just in case we've changed the orientation, and click Save, and it should produce your AGP file. Okay, so once we have our AGP file, we can start to produce our new assembly based on the changes that we made in the pretext map. So if we cd into our test data, oops, we should find that we have our AGP. What we need in order to able to kind of reconstruct a new FASTA file is we need to produce a TPF file from our assembly file. We do that with rapid split. So Perl, the script is in the directory above. So, and if we just hit enter, it should give us a little usage. As you can see, we literally just give it the FASTA file. So -FA and our assembly file. And in a very brief moment, that should have produced us a TPF. And there we have it. So what the TPF is literally a flat text file that converts all of the sequence into a line of text that says we have essentially a scaffold, but it will but contigs. So it will be scaffold 1 from X coordinate to Y coordinate, followed by a gap line indicated by which ends in our scaffold, followed by another line that says scaffold 1, and then another coordinate based on where it starts and ends. And this is basically just a way of producing a file that can be worked with very, very quickly and is very light on kind of resources. So once we have a TPF file, and now that we've got our AGP file, we can convert this into a reconstructed TPF file of our changes. So we do that with pretext TPF, which is run using PTT in this workspace. Minus help, we can have some options. There we go. So we want to use -A and give it our original TPF file from our original assembly. We want to use -P and give it the AGP from pretext, and we'll give it an out file, and we'll write a log. We'll write a log. This will tell us how many changes we made in reworking our genome, how many autosomes, whether we've tagged sex chromosomes, amount of sequence that we've decided we're going to remove as haplotigs, etc. If you need to kind of report what you're doing to supervisors or managers, then this is a way of showing them what kind of work how much work you actually done. So anyway, so PTT -A, and we want to give it our original pretext, our original TPF, dot, dot, and our AGP. Oh. Our out file, so that's out file. And we'll write the log. And there we go. In an instant, we have our new set of files. So we have a new TPF. So this is our TPF with all of the changes that we made in the AGP. We have another TPF here, which is all of the sequences that we want to remove from the assembly because they are false duplications, and we can generate the FASTA file and merge that with the alt assembly that is produced through the assembly process. We also have a CSV file, a chromosome CSV file, which tells the next script the sequences that are going to be designated as chromosomes, and if we've identified a sex chromosome, it will also designate these as sex. So it will write that these are an X or a Y or a Z or a W, etc. And also we have a log, and if we look at the log, it will tell us just what we've done. So it gives us a resolution. This is how many base pairs are in a text cell. I think there was a question in the pretext about how accurate placing a sequence is if we're not trying to import a sequence, place a sequence in a gap. And this is in this genome, this is how accurate this placement is, within plus or minus nearly 10,000 base pairs. It will tell us, so we have one sex chromosome Z, and we have 30 other autosomes. We have a number of other unplaced sequences, and we're also removing 5 sequences that were haplotigs. And at the end here, we made one contig. So that was an actual physical break in the sequence. So that was a contigging error by HiC, plus 4 breaks in gaps. So these were scaffolding errors by YARS normally, but SALSA or 3D DNA, whatever scaffolding you're using. And we made 29 joins in the sequence. So in order to reconstruct our new FASTA file, we are going to use something called rapid join. And again, we can get some usage out of this. So this version only copes with a single haplotype at a time. As I said in the chat, we're currently migrating everything off of GitLab over to GitHub. When everything goes up on GitHub, and I'll spam various channels once that's up. Once this goes up on GitHub, there will be an updated version of the script that will allow you to produce multiple haplotype files at once. So if you're working with a diploid genome, you could produce haplotype 1 and haplotype 2 FASTA files at the same time. But in this case, well, we've only got a primary, so this will suffice for the moment. So again, we need to provide our original FASTA assembly file. We will provide our new TPF file, our chromosome CSV, our list file designating which sequences are actually chromosomes, and we'll give it an out file prefix that will kind of append a prefix onto a FASTA. We'll also run it a second time to produce the haplotigs FASTA as well. So Fingers crossed we have enough resource to be able to produce the assembly. It should only take 30 seconds, a minute or so. In fact, there we go. So what we have produced are these two finalized files here, workshop chromosome list file and our assembly file. What the list file enables, so this is what you use to submit with your genome to ENA or to NCBI, and this lets them know which sequences you are designating as chromosomes. So when you go to your GCA on the page on NCBI, you'll see a list, and you'll see the table at the table on the assembly page. It will basically list all the accessions for each of the sequences that are designated as chromosomes. If you don't include a CSV file, it will just go in as scaffolds, and we'll just get a standard WGS accession. So that's literally it. Oh, we can generate the HAPs as well. So we do not need the CSV. We do want the haplotigs, and we want the -hap. And that will produce even quicker. And here we have our additional haplotigs file that we can then merge in with our alt assembly file to ensure that we're not actually losing sequence from the assembly, because there is value in the alt and people will use the alt. And actually nowadays people are more and more producing the 2 haplotype kind of resolved assemblies. What can I do? We can kind of look to see how many sequences we've got in actually, I don't even know if rep's installed here.
It should be.
Yeah.
So we can get a list of all the sequences that are in the file, and you can see that we've actually renamed our sequences. So we have Super Z, and then we have our 30 other autosomes and a bunch of other unplaced scaffolds. And that is about it. Does anyone have any last questions regarding the process that they've seen? I guess not.
Somebody asked on the Discord, Joe, if you had like examples for generating the track data. I haven't got any yet, but I did point them towards curation pretext as another way to do it. But
we can provide you, we can kind of point out how to generate some of these, and I'll put into the Discord. So there is 2 pipelines available that are both public that will produce all of the necessary files. The NF core, Damon?
Yep, they're both NF core.
Yep. So they're NF core pipelines, Dockerized, so you can run them without having to install a ton of stuff. They will produce the HiC contact maps with all of the files embedded. So that would be telomeres, coverage, gaps, and repeat densities still there.
I want to say yes.
And if you run the kind of, so if you run the tree-val pipeline that Damon ran a workshop on a couple of days ago, then that will also produce you, if you want it to, it will produce you a sequence browser to enable you to kind of interrogate your sequences in another way. This will give you a basic linear browser view with gene annotation attached to it, as well as all of the information that we would use. So you can embed the HiC data onto that. You can also embed the gaps and telomeres and so forth. What you can't see at that point is how the HiC interacts beyond the boundaries of a single scaffold. But you can at least verify the integrity of each individual scaffold. You can get base resolution on that. But typically, we find that for the majority of cases that just running the HiC contact map is enough information. But either of those two pipelines, the TreeVal or the curation pretext, and I'll post both of those into the Discord channel, will allow you to produce all of the files necessary.
Cool. Thank you very much. Right, we are going to have to end it there, everyone.
I hope everyone found it useful.
So I'm amazed every time I watch this, in all honesty. Magic. Yeah, if anyone has any other questions, please use the Discord. And I'll see you in another session. Goodbye.